That must mean it's wanting a lot of protection, and it must be really hard to get in and out of that structure. So what ways would you have of actually getting in and getting out? Well, for right now, the most obvious ways, if you look right there where my laser is pointing, these little flower-looking like things are called rosettes, because that's the pattern of proteins. But they're pores, they're an actual hole, all the way through the membranes, both of them. That's how you can get in and get out, if you're small enough, if you're given permission.
If you have a trans.
Because what's really cool is those pores, imagine then, I guess, having octopuses sitting on the edge of the entrance and putting their tentacles over the opening, like guards. So not only is there a hole there, but they're also kind of protected by the proteins that kind of make sure that you actually should be going through the tunnel and getting inside. Well, what is so daggum important inside the nucleus that it has two membranes and kind of guards around their pores? This is where your DNA is kept. Remember, this is one of the things that separates eukaryotes from prokaryotes. Our DNA is in the nucleus. And your DNA is where your cell has, that's basically your cell's central computer, where it knows how to do anything that it needs to do. That information is stored inside that.
So you said the rosettes were protein, or they were protected by protein?
No, the proteins are arranged in a flower-like pattern.
Okay.
So if you look at each little bump there, each one of those little bumps is an actual protein that goes through the membrane, and they form a tunnel all the way through, and it has a rosette pattern.
Okay.
All right, now let's look at that a little more closely. In the center of the nucleus, there's no membrane around this part. It is just simply a region where there's condensed, morely packed DNA. It's called the nucleolus. So the nucleus, then you have the nucleolus. And the nucleolus is found within the nucleus, and that's where most of the DNA is kept. We can also get another picture of looking at the fact that this nucleus has a double membrane structure. And until this becomes automatic to you, you want to kind of make yourself go through these steps. So you see that line right there? That is one plasma membrane. But we can see how it's folded back on itself to give us the other plasma membrane. So we do have two membranes that are there. Also now notice that there's another structure we're going to tell you about, known as the endoplasmic reticulum. Right? That's going to be this kind of blue folded looking like stuff right here. And as I'm going to try and teach you guys to be in the habit of, really study the picture for a moment. And if you really study that picture, you can see, all right, I can see my two membranes. I can see my pore. I can see the tunnel that John was talking about. Now, if I follow this membrane right here, if I literally look at it and understand what's going on, this membrane becomes the endoplasmic reticulum. It's an extension of the outer membrane of your cell's membrane. Then I look at the endoplasmic reticulum, the blue folded part, and again I remember and I reinforce the fact that that blue line, or that light blue line, is actually a representation of a phospholipid bilayer, which means it is semi-permeable, which means you're going to have protein stuck in it. What proteins does it have stuck in it? These guys, called ribosomes. These will make proteins. We're going to learn how they do that a bit later, not today. So ribosomes make proteins. That means then that the endoplasmic reticulum is also a place where proteins can be made inside of your cell. Remember, today's Wednesday. Monday we kind of looked at, but definitely last Friday. We talked about one of the differences between cells, and we mentioned something called the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Since this is endoplasmic reticulum and it has bumps on it, it has a rough looking surface. So this is actually rough endoplasmic reticulum. All right? And it means rough endoplasmic reticulum makes proteins.
So is ribosome a kind of protein?
It's a protein, because remember, most things in a cell are protein anyways. But it's going to be a protein that's going to play a role in making other proteins. It's sort of like saying, in a steel factory, right, there are steel machines that make steel, because they're made up of steel.
So if there's a smooth endoplasmic reticulum, it does not make protein.
Like that right there? Exactly. All right? So the next organelle, away from the nucleus, you want to kind of think about the order, the path, that if you were in the middle of the nucleus, you're going to walk through a double membrane structure, end up inside the endoplasmic reticulum, known as the rough endoplasmic reticulum. Right? But then you keep on going. And you can see all this stuff. It looks like sandpaper, so it has a nice little rough surface to it. And here they simply have just removed the nucleus so it doesn't get in the way of the image. But now we have out here the smooth endoplasmic reticulum. They play a role in making lipids, fats. So you have the nucleus that contains DNA. You have the rough endoplasmic reticulum that plays a role in making proteins. The smooth plays a role in making lipids. One of the best ways, boy, okay, maybe I do need more than 4.5 hours sleep. One of the best ways to think about, I'll go for 4 3/4 next time, is put your hand over a cooked steak. Does it feel rough? Yes, it does, right?
Yeah, it is.
Steak feels rough. Steak is protein, made in the rough endoplasmic reticulum. Now rub your hand over butter. Does butter feel smooth? Butter's a fat, maybe a smooth ER. All right, if that helps at all.
So rough endoplasmic reticulum makes proteins,
Yeah.
Mm-hmm.
and then the smooth one makes lipids?
Mm-hmm.
Okay.
All right, now we'll save that word for the next little picture coming up. The next organelle we're going through is called the Golgi apparatus. It is not actually directly connected to the ER, to the smooth ER, or to the rough ER. It is the third thing in order, so it's after the smooth ER, but it has what some books call a stack of pancake appearance, where if we make, I guess we'll make this the nucleus, and then we'll just sort of go like this and make this our ER for the moment. You're going to have an organelle that has layers of membrane structure, but they're kind of folded like this, like hotcakes or pancakes. And whatever comes off of the smooth ER is going to go towards the Golgi apparatus. But it goes through this layer first. Then it goes to this layer. Then it goes to that layer. Then it goes to this layer. And then it'll finally leave after that layer. You'll learn way more about this in Bio 211. But the idea is that this Golgi apparatus, I'm trying to show you that right here, see where this is the part closest to the nucleus, and then stuff going from the ER to the Golgi apparatus will go in on this side, and by the time it's done, it'll leave on this side. Well, how do you get from the smooth ER to the endoplasmic reticulum? Remember the inside of a cell has lots of water in it. It's called cytoplasm, right? And we know we have hydrophobic and hydrophilic things in biology. You know, the hydrophobic cannot be transported very well inside of water. So far, so good? And the cell is mainly full of water. So how do you get hydrophobic things, like lipids, to be transported in a, quote, aqueous or water environment?
Assistance.
You have to put them in a special taxi cab. You have to isolate them inside of a structure that is hydrophilic on the outside of it. That structure is called a vesicle, called a transport vesicle. Right? So you're going to be transporting stuff from one part of a cell to another, but it's a vesicle. What's a vesicle? And not a vesicle from Star Trek. A vessel. If I took my grandfather's ashes and I put it in an urn, is an urn a vesicle in a sense?
Yeah.
Yeah, right? It's just a container that holds stuff.
Oh.
Right? So grandpa doesn't go flying all over the house. Well, look at the structure of our transport vesicle. Does this structure here look the same as that structure right there?
Yeah.
And
Yes.
you're gonna say yes.
Yeah.
Okay. And does this look anything like this?
Yeah.
Yeah. And if you even look here in the previous slide, right? That's a vesicle. It's a transport vesicle. It looks the same color and the same basic idea as this part over here, because it's made of the same stuff. What happens is the end of the smoothie AR is going to pinch a part of itself off. Like if you had a big ball of dough and you just pinch a little piece off of it, that little ball of dough was a part of the bigger ball, right? Well, this was a part of that. And since that is a plasma membrane, then that has to be made up of a plasma membrane as well. They're both made up of phospholipid bilayers, which makes sense because then the outside of my phospholipid bilayer is hydrophilic, right? Because of all the charged phosphates. All you have now is a big hollow sphere, a ball, and in that ball, the cell has put stuff in it that came from the smooth AR or the rough ER. That's what those little green balls represent. It's whatever came from the ERs, the smooth or the rough. Now, since it's made up of because it's made up of a plasma membrane that was the structure of the smooth ER or the rough ER, it can then merge and literally melt and become a part of the Golgi apparatus because it is also made up of a phospholipid bilayer. Right, well, think about that for a second. Anybody confused?
Can you just say go over
Go over it. What is it?
So the idea that
So let me ask you this.
yeah.
What is a nucleus? What is the nucleus made up of? It's the
DNA.
structure, the outer wall.
DNA.
But what's the outer wall made up of?
Phospholipids.
All right. What is the wall of the rough ER made up of? What is the wall of the smooth ER made up of? So now what is a vesicle made up of? Same stuff.
Oh,
And that's because the ER is going to just pinch its part off.
and make.
Because I want what I just made to get to you. And you're the Golgi apparatus, I'm the ER, but there's all this stupid aqueous stuff in the way. And this is hydrophobic. I just made a fact.
Oh, because they're not connected.
Right.
ER.
So I need to transport it to you, but I need to put it in a vesicle that can move through water. So I pinch a part of myself, which I can do that, okay. Pinch a part of myself off, now it's surrounded by a plasma membrane. And now it's going to go to you and become a part of you. It literally melts and becomes a part of your structure. And when I do that, I release what I just made into you.
So what is made, like what is being released? Is it just
Proteins or fats, whatever. If you came from the rough ER, it'd be a protein.
Okay.
If you came from the smooth ER, you would then be a lipid. Which is what they're showing right down here, in a sense.
And phosphols and or lipids and proteins can't be transported without the vesicle.
Definitely lipids for sure, right? But some proteins are have polar charges to them. They could dissolve in water nicely, but I want to make sure that they get to their final destination. So what I think about is the rough ER and the smooth ER and the Golgi apparatus are all factories with one goal, to make a product. And each part of the cell makes a part of the overall product. Like an auto factory, right? One part makes the engine, one part makes the transmission, one part puts the wheels on, one part puts in the stereo, and all that stuff. And I need to transport all the things that my department makes to the assembly portion, so they can put it all together. Well, the Golgi apparatus is kind of like the assembly region, where the ERs make the parts. I need to put them together in the Golgi apparatus.
Okay.
So the transfer vesicle becomes a part of the Golgi apparatus?
Yep. Anytime you have similar structures, like dissolves like, they can become a part of each other. All right, so here's this transport vesicle. It merges into this end, and what's going to happen is whatever goes in there is going to be modified. It's going to be altered, refined. And by the time it's done, it's going to come out the other side, but you're going to be facing more cytoplasm. So what do you have to be put in? Another vesicle. So we go from the ER via a vesicle to the Golgi apparatus. It does its thing, and when it's done, it pinches a part of itself off. See right there,
Oh,
we're
it's
pinching ourselves off.
side.
And now there's another transport vesicle. And this vesicle is going to go to the cell's plasma membrane. And since this is a plasma membrane, and the cell's membrane is a plasma membrane, and like things can merge together to form like things, when I go up to the cell's wall, I'm a vesicle, and we'll make the wall, the membrane here. I'm going to come up to the membrane, and I become a part of it. This part of my hand joins that part of the membrane. This part of my hand joins that part of the membrane. And I just spit out what I just made, and that's now released to the outside world. But we need to name these things. We got names for everything, don't forget. So let's do first this part right here, because it's a little more bigger and visible. So this right here, right, is the rough ER. Right? There's my ribosome. Remember, the ribosomes are attached to the rough ER. And we saw that in the earlier picture. We go back to here, we can see the little dots right there. And if we go back to here, whoops, come here. We had another, there we go, that was what I was looking at. We can even see them back on this one, right? So they are attached. There are proteins stuck to the surface of that membrane structure. Well, how they do it, don't worry about it right now, but here's the ribosome making the first part of a protein. And this is where you always want to pause and draw back on what you know so far. Proteins. How many different layers of organization and structure do proteins have? Okay. What is the name of the first layer? Okay. How would you describe the primary structure of a protein?
It's like the amino acid, a polypeptide all
But-
together.
Now, what's its shape look like?
It's like this.
What's this?
It's a length along one line.
One line, like a snake
Yes.
or a noodle.
Yes.
Right?
So,
Is that a snake or a noodle? Is that a
Yeah.
primary structure of a protein?
Yeah.
Bingo. What's the secondary structure look like? Is it folding? Right? The first layer of folding? And what's the tertiary? More folding on top of folding? Tertiary. So what is the Golgi apparatus doing? It's folding the protein to the proper shape. It's modifying the protein so it has the proper shape. Is it a 2-layer cake, a 4-layer cake? Is it a wedding cake? Is it a birthday cake? That's what the Golgi apparatus does. It just provides the environment for the proper folding. There's no little workers inside to actually do the folding. It's all chemistry. But it has the right environment for the chemistry to work.
Does it go to the quarantary state in the Golgi apparatus, or not?
Sometimes it does, yes, sometimes it doesn't. Now, you're the Golgi apparatus, you just got done making the cake, you're a baker, you need to now deliver it to the party. So it has to go in the van, right? So you can transport it across town. Our vans are vesicles. Well, how does a vesicle get made? So we can see, right, plasma membrane. Plasma membrane. What does this picture try to convey to you? It's pinching a part of itself off. That part is called endocytosis. Endocytosis means the creation of a vesicle, pinching something off. Now my vesicle is a ball of phospholipid bilayers that contain something inside of it. Maybe it's a protein, maybe it's a lipid. And when you now look at the organelles of the nucleus, the rough ER, smooth ER, and Golgi apparatus, that creates what we call the endomembrane system. Endo for within. It's the within membrane system, because they're all made up of cell membrane material, meaning a phospholipid bilayer. And they're trying to show you the path of how stuff could go from the rough ER to the smooth ER to the Golgi apparatus. There's my endocytosis, and then it's released. What happens to it? Well, it could go to the cell membrane and become a part of the cell membrane, or it can go to the cell membrane and be excreted or kicked out of the cell, because maybe some other cell needs what that cell just made. Or it could do something else within the cell. It all depends upon what that is. But we need to give you one more word. So here is the vesicle, right? It is a phospholipid bilayer containing stuff made from maybe my ERs. It's going to merge with the first part of my Golgi apparatus. See how they're trying to show you how it's merging, became a part of it? Right? So, in a sense, this is going to become a part of that.
Globally layered.
Yeah, let's save that word for later. Okay. Now, when we get to that end, we can look a little more closely on how that can actually happen and why you want vesicles. So we look at this picture here and we see a plasma membrane, and so we always start and go, that means a phospholipid bilayer. There's a hydrophobic part, a hydrophilic part, there's a thick lipid part. It's semi-permeable, it's restrictive, it has proteins in it. I mean, just do that every time until it becomes just one big thought in your head, like a reflex. All right, so what are they showing me here? Well, here's a blob of something. Maybe it's nutrients that the cell wants. Remember, it can't just go through the cell wall. It's selectively permeable. So how can this get inside the cell? Well, it approaches the cell's membrane, and then the membrane begins to sort of like yield and back up and create a pocket for the stuff to go into. And then you finish pinching off the pocket. So then when this part pinches off, it becomes what's called the food vacuole. And this structure, what is this made up of, do you think? Cell membrane, right? Which means the outer layer of it, the outer portion of it, is then going to be a hydrophilic layer, which means it can travel well in water, and the inside of a cell is made of water, so this is a nice convenient structure. Now, over here, we have another vesicle, but it has enzymes in it. For right now, think that enzymes can help break things down, dissolve them. A way for you to realize this is put a cracker in your mouth this afternoon. Don't swallow it, just let it sit there. All right? It's going to dissolve, right? Because of the enzymes in your saliva. Well, because they're breaking down the cracker. These enzymes need to get to the food vacuole. Well, how do they do that? Well, this vesicle containing digestive enzymes is going to do what according to the picture? It's going to bump up to the food vacuole, and since they're made of the same thing, they become one big lobular structure. But now I've exposed the food vacuole to my enzymes. And then they're going to break it down, and then the cell can do whatever it needs to do with that food particle.
Where did these digestive enzymes come from? Are they just...
Well, they're proteins. They're probably going to be made by the rough ER and pitched up by the Golgi apparatus. If you go back to this, it was a very nicely timed question. Various locations in the cell. So maybe that thing contains digestive enzymes. And then it just has to know what to attach to, and there'll be proteins on the outside that it knows what to do and where not to do it, in a sense. Membrane structures. Well, here's another one. Here's a vesicle. That means a spherical ball made up of the cell's membrane-like material. right, containing damaged mitochondria. For right now, just think of this. The mitochondria are a factory inside of a cell. The factory is now old. So if you are from, let's say, Jersey or New York, and there's a really old building, what are they going to do to the building? They're going to break it down, right? And then in today's technology, you're going to go through and take out all the steel and use the steel, maybe reuse the bricks. Well, here's an old factory that's dying in the cell. A lysosome. What is the structure of a lysosome? Cell membrane, right? It's going to merge with another vesicle that's containing, surrounding this old dying factory. It has enzymes that will break down that dead, dying factory, merges together, so that I can reuse the parts. Your cell is an amazing recycling center.
It says recycling.
It just keeps recycling its parts, because the proteins that made this could be used for something else maybe. Maybe making another part of a receptor on your cell's surface. Right? But what you have is a vesicle containing digestive enzymes that I could use to take in nutrients and get the nutrients I need for the cell to work with. Or if there's a part of myself that's breaking down, no longer functional, I can break it down and reuse its parts by now using the lysosome that contains enzymes. Cell membrane, cell membrane, cell membrane, cell membrane, cell membranes. That's all you got to keep in mind, so it makes a little more sense to how this can happen.
So after it goes through the digestion process, does the cell membrane just get absorbed by, the other cell membrane? Like, is that part of the
Which cell
recycling?
membrane? This one right here?
Yeah, does that get absorbed by the gray cell membrane?
It depends on what that is. So if it is something that could be recycled, then there could be another organelle that'll come around and simply just break that down and say, everything in here is okay, we can actually release it inside the room. And then those parts can float around and be used whatever they need to be used for.
And another question, so are these floating around outside the endomembrane system?
Yes.
Just like
Right.
randomly?
So if this is my nucleus and my arm is the cell's membrane, they're floating all through here.
Okay.
All right, well, let's look at those things called mitochondria. And we're going to add more detail a bit later. They're found in almost all eukaryotes, right? That means we have a nucleus, right? We have a cell membrane, we have organelles, we have structures. Mitochondria have two membranes surrounding them. Okay, I can see that, right? There's one membrane here. The inner one, you need to realize, is highly folded. That's important to the structure of mitochondria. The inner membrane is highly folded. Now, most people learn that mitochondria are called the power plants or the powerhouse of a cell, because they make the one thing that you need to survive with, called ATP. That is the token, the energy, the currency that cells have to use to do things with. So if it requires energy, that is the form the energy comes in. This is the fuel for your cells. And we'll learn about its structure a little bit later. Now, cells have lots of mitochondria. And different parts of your body have different amounts of mitochondria. For example, when you get into 241, you'll learn about how skeletal muscle cells. Every muscle cell has a fair number of mitochondria to them. But cardiac tissue has way more. Because if cells need energy to do something, ask yourself that when was the last time your heart took a half-hour break?
Never.
Right? That means it has a constant need for energy, which means I need lots of power plants within that cell to make sure I don't run out of energy to do whatever I need to do. All right? Well, plants have mitochondria, but they also have something called a chloroplast. How many membranes does a chloroplast have? Take a look at the picture. What do you see?
Two.
Exactly. Two membranes, right? Two phospholipid bilayers. Two semi-permeable structures. Now, inside of it, though, it also has what looks like Oreo cookies or poker chips. that are stacked up on each other, right? The collection of them are called granum. Any one individual cookie or poker chip is called a thylakoid. Thylakoid. So you got to practice spelling, right? Granum and thylakoids. So if I were to ask you what two structures are found within a chloroplast, you would say, well, individual thylakoids are stacked up on each other to form a granum. And there are several granums, right, in a chloroplast. Then for right now, does anybody know what chloroplasts do? So what does that do?
Absorb
No, absorb what?
energy.
Like,
Or absorb sunlight to create energy.
But isn't sunlight energy?
Or is it in plant language?
Okay, sure, that works. All right. So a chloroplast does what's called photosynthesis. We'll learn all about that later. But what does it really do? It turns sunlight into food energy for the plant to use. So it turns sunlight into food energy.
That's what the thylakoids.
That's what the chloroplast does.
Oh, the chloroplast.
Right.
Okay.
It'll use the thylakoids to do that with. So, and then you eat the plant, which has the food in it.
glucose. What exactly is a stroma?
Stroma is the area around the granums.
Stacked up.
If you look at, for example, mitochondria, they have stroma as well. It's the space in between the membranes where you see those little green dots. So where I am not standing in this room, and where there are no tables, that's the stroma. All right, well, here's our big question. All right, I'm gonna pick on previous students of mine who have not had Bio 160 before, but have had other classes, to see if they know the answer to this. What is unique about mitochondria besides making ATP and being a double membrane structure? You guys remember? I don't hear any voices. Well, I hear voices, but not the ones I want to hear.
There's a ribosome in the mitochondria.
No? So,
The DNA in there.
let's say there's been a severe plane crash and the individual victims are unidentifiable by their facial structure because it's been all burned, crushed to pieces. How can I tell that it's still you? How do I identify you?
The
It's
DNA.
a
DNA, all right? Because we know your DNA and every one of your cells is the same.
Yeah.
And that you got your DNA from mom and dad. Mitochondria have DNA, but it's not your DNA. It's bacteria DNA. Yeah, it's a separate DNA than your own. It's bacteria DNA. Which means in every single one of your cells that needs mitochondria, living inside of it is an ancestral bacteria organism that makes ATP. So if you want to know your ancestry, then you're going to do what's something called mitochondrial DNA. I'm writing out a room here. Now, we'll circle that so it's separate. Oh, I just made a mitochondria. Cool, okay. All of my mitochondria only came from one of my parents. It was my mom. Dads never contribute mitochondria DNA to their offspring. So when you check your ancestry, I'm going to follow your maternal pathway. Your mom, your grandmother, your great-grandmother, your great-great, your great-great-great-great, your great-great-great. I can go all the way back, and we have gone all the way back to understand that more than likely, all of us, well, not maybe all of us, there might be 2 of you that are excluded from this, came from a village on the eastern portion of Ethiopia. That's where your ancestor goes back in time.
Yeah, we all came from Africa.
We're talking 200, 300, 400,000, 500,000 years ago.
About, except a couple of us that might have like Neanderthal DNA.
No, they came from there too.
Yeah, everybody came from Africa.
Everybody, well.
Not everybody.
Oh, yeah, no, that's true, yes. But not everybody came from the portion of Ethiopians. All right, there might be some true Nigerians or Somalians that their family never actually left Africa. But everybody else that's not African, you came from Africa.
So you guys are all
You're
African
African.
Americans. You just don't know it.
So we're all, exactly, well, we're all African humans, in a sense. All right, and we'll talk about why that should be. Take a quick glance just around just for a minute, just look at everybody's faces for a moment,
Yeah, I learned that.
and
in
you
first
realize
class.
there are various shades of color in the room. And we could line you up from darker to lightest. All right, and that tells us basically where your ancestors lived over a long period of time. Well, how was this able to be done? There's a geneticist from Stanford called Spencer Wells. And he thought, I want to figure out where we all came from. That's a big argument in biology about, the waves of migration from wherever we came from. So he went through all the world and took samples of people and looked at their mitochondrial DNA, and in big genetic important languages, it doesn't change much. But I can then tell when it does change, and I can work my way backwards through time. Because all of your cells, all your mitochondria came from your mom. Because this is actually a bacterial ancestral organism living inside of every one of your cells. So the big question in biology was, how the hell did it get there? I mean, what's a bacteria cell doing living in one of my cells? I'm not a bacteria, I'm a eukaryote. What's a prokaryote living in my cells?
What'd she say?
So here you have a cell. One of your great ancestors. Now, let's go back 3.5 to 4 billion years ago. It is a mean, cruel, harsh environment. There's no atmosphere in terms of oxygen. It's a pure CO2 atmosphere. There are volcanoes going right and left. There's solar radiation, high acidity in the water. It's not a very friendly place to live. Well, here is this organism, the bacteria, that went to maybe attack this cell, and it got swallowed up by the cell. Well, we know how that happens, right? It's the same way as a food vacuole works.
Just
Right?
combines.
Here comes that bacteria, and they got swallowed up by the cell, thinking, I'm going to eat you for breakfast. Right? I'm going to destroy you. And for whatever reason, that did not happen. Well, that's where that second membrane came from. It only has one membrane, but they really should show just one. Then it gets inside the cell, and it might be that that is where the other membrane came from. They're still trying to figure that part out, so they need to do some more studies on the outer membrane. But the idea is that for whatever reason, this cell either could not destroy that, or it didn't see it as a threat. And now this thing is living inside of this cell. It's protected from the outside harsh environment. And it just so happened it started spitting out tokens. I mean, if think of it this way. You had a total stranger come move into your house. They started throwing $10 bills out all over the place. You're going to kick him out?
No.
You're going to keep and collect the money, right, and use it to this is money.
That's how my auntie is.
This is what your cells need to survive
Just
with
keep
and to
throwing
work
money,
with.
baby.
So what you have then is a symbiotic relationship. Two things living together, each one helping the other out. The host cell is protecting the little cell, and the little cell is spitting out energy that the big cell can use. And since it's happening inside the cell, that's what endo refers to. Symbiosis means living together, so living together inside. And that's how we think mitochondria ended up inside of your cells. It's doing the same thing for chloroplasts, for plants. That's our best, strongest hypothesis in terms of how they came to be together. Well, we mentioned that cells have a skeleton a while ago. It's called a cyto for cell, skeleton. There are three parts to making up your cell skeleton. three different protein structures. One is called the microfilament. Think about your hair. It's a filament structure. It's a long cylindrical thing. Micro means small. So it's a small thread-like thing. It just so happens to be made up of little round proteins that are stuck together, and there are two strands of those little round proteins. So imagine two strands of pearls, and you just twist them on each other. That creates a microfilament. Then you have intermediate filaments. Look like copper wires that are wrapped around each other. They're bigger, they're stronger than a microfilament, because micro means small. Inter means in between, in size. But then you have a micro, which means small. What's a tube?
It's like
It's a round tube. A tube is a round tube. Okay? It's a round, circular, hollow thing, right? Is this round and hollow? No, it's a solid object. That's not hollow either. So this is a teeny filament. This is a teeny tube inside the cell. Now they all have different functions and different jobs. Yeah, we got time. In a quick thing, we're going to come back to this on next lecture. I want to show you a bit of a video. So we're going to go fast here for a second. Microfilaments change the cell shape. Microtubules are the railways, the light rail system inside of your cells. We're going to see some pictures of this. And the intermediate filaments help to keep things held in place. There's a really cool video I want to show you a clip of that was made through Harvard University after years and years and years of study. What you're about to see is not fictitious Hollywood. It is what happens inside of your cell to the best of our understanding on a micro level. So this cell is going to be like the size of this building. And you're going to be inside the cell looking at it to see what it actually does, or how it does what it does. This is what's called Inner Life of a Cell. You can find them on YouTube in various portions. Some are narrated, some are not. This up there is inside my cell. That's my cell membrane, and these are proteins anchoring this thing to the cell's membrane, so it doesn't go away, it doesn't move around. So you're inside the cell. The ceiling is the inside of the cell membrane. You're now on the outside of the cell. I'm sorry, you're on the inside looking down, but you're actually looking at the upside of the cell membrane. All those little things are phospholipid heads. This is what's called a raft, transporting proteins from one part of the cell to another. We're just upside down. So look at this structure down below here, right? You're looking at a phospholipid bilayer, right? See how it wiggles and jiggles? That's a fluid mosaic model. To go back and emphasize that, to show you how good they were when they made this. Okay, I want you to follow that green phospholipid. Realize it doesn't stay there. They're all moving around. You'll learn these in 211. There's a protein receptor. So, top part of the screen is the underside of a cell membrane. These are intermediate filaments. You have other protein parts of your cytoskeleton over here. So these would be microfilaments up here, excuse me. Those would be micros, and these are intermediate filaments over here. Transmembrane protein. Don't worry about that. Let's get to that's the cytoskeleton. You're like a diver diving deeper and deeper into the cell. See how crowded it actually is. Okay. This is a tube. It is a microtubule. They're showing you how they're actually made. And in 211, you'll learn what's called treadmilling process. The actual way you make a microtubule. Now microtubules play a role in transporting stuff from one place to another. Here's one end breaking down so I can reuse the parts to make another microtubule somewhere else. And that's what I wanted you guys to see. Whoop, let's let the words show up. All right, that big blue thing's a vesicle. What's it made up of? A phosphorobilayer. This is a microtubule. This little dude is called a motor protein. He's actually walking along the microtubule, and he's carrying a vesicle to wherever it needs to go. It's like a little mini person in a sense. And every step requires ATP.